Evolutionary biology makes many kinds of contributions to the health sciences. For example, it helps illuminate the origins and the ongoing adaptations of infectious diseases, a subject of particular interest to us at the moment. It helps to explain the extent and patterns of human genetic variation in resistance to diseases and other kinds of traits. And it explores other medical implications of human evolutionary history, including adaptations to former environments. So let's start with the pathogens and their evolution. We've already, some weeks ago, it seems like years ago, doesn't it, considered the evolutionary history of HIV, the human immunodeficiency virus that is a cause of AIDS. Where did it come from? And we presented then the evidence that it came from old world monkeys and apes. That's where they're found, so it's obviously that's where they've been longest. Several kinds of phylogenetic analysis have shown that HIV-1, which is a particular group of closely related lineages or strains, and they're the virulent ones that cause AIDS. They jumped from chimpanzees to humans more than once. So this tree makes that pretty obvious. So here's a chimpanzee form, here's some others, here's another one down here, the sister group to all the others. So this pattern clearly implies that this group O of HIV-1 strains must have come from chimps. Similarly, this one, YBF30, the N group, came from chimps. It's just embedded deep in a large group of chimpanzee lineages. And similarly, the diverse M group, which is the one of major medical importance, since it's by far the most common one in humans, it also clearly came from chimpanzees. That's something we could not have known without evolutionary phylogenetic analysis. It also, a subtler point is that this tree, its shape, shows that the M group expanded very rapidly following its entry into the human population. So its ancestry is does not have a flat, shallow genealogy, but the M group does. There are many short branches deep in the tree of the M group, followed by proliferation of tips from throughout that tree. That pattern is best explained by, really demands a very rapid expansion in numbers and probably geographic extent following this node, which represents the place where it entered humans from simians who appear to have been chimps. When did it arrive in the human population? We also saw this story some weeks ago. This is just to remind you of it, and the original short and classic publications, especially this science paper, which I urge you to go read if you have time and interest. Bette Korber and her colleagues sequenced the envelope protein genes in a large number of viruses from this group, and then inferred their evolutionary history by building phylogenetic trees. This one on the left is an unrooted maximum likelihood tree, but they were able to root it by using outgroups, and then used the dates when these very diverse strains had been collected to estimate the rate at which the envelope protein gene evolves. So this is just a scatter plot showing the year the sample was collected, and then in color and with the letters, the subgroup it came from, and then its divergence from the common ancestor, that orange spot on the tree in the upper left, and then they fit a line through it, and whoops, let's go back, then extrapolated that line down to 0 divergence from the common ancestor in order to date the common ancestor, and achieved this wonderful result, which is that with high confidence, the ancestor must have lived before 1950. So it ruled out origins after 1950, which meant, among other things, that it did not arise, it did not get into the human population from polio vaccine tests which had taken place in Africa involving chimpanzees, which was a sort of a conspiracy theory that was popular for a number of years. This is an example of phylogenetic analysis, real evolutionary biology that decisively rules out a hypothesis that was attractive to many people for where it might have come from and how it got into the human population. So influenza, the other really big killer, where does it come from? Here again is this picture, this cartoon of what influenza virus looks like with these hemagglutinin and neuraminidase spiky proteins on the outside and its RNA genome in 8 segments in the inside and so on. It has 11 genes for polymerases, structural proteins, and then 2 for these coat proteins. Here's the hemagglutinin, a space-filling model of it, and the colored amino acid residues show sites that are seen by our immune system and interact with our immune system. The flu phylogeny is often constructed based on an internal structural protein that does not interact in this highly unstable way with the human immune system, so it's thought to be more reliable as a way of inferring history. And what this rich and complex phylogeny reveals is frequent transfers between different host species, which include horses, pigs, and several groups of birds, especially gulls and ducks, turkeys, chickens, and so on, as well as humans. And in addition, all the strains are quite closely related. So because we have dates for all of the samples from all of these species, including us, it is possible to reconstruct the story of how it jumped between different species of hosts. So here were clicker questions last year. We can just walk through them. Did these strains that were collected from pigs and are on these short red branches, did they jump from humans to pigs or from pigs to humans? Well, these yellow branched ones are all more or less famous human strains, including H1N1. No, so they went from humans into pigs, so pigs got them from us. Here's a group of swine strains that have stayed in swine, except for these two. Yes, here's the 1976 H1N1 in humans, and here's the 1988 H1N1. Did they come from pigs into humans or from humans into pigs? Well, the correct answer is from pigs to humans. Obviously, that's a much more parsimonious reconstruction than the other way around, because they're the closest relatives on all sides are pigs in both cases. The super deadly 1918 strain, which you've probably heard of in the last few months or weeks, even if you never did before, it was a major factor in World War I, for example. It's near the base of the modern human swine clade, so-called. Here are a bunch of swine strains, and here are a bunch of human strains. But its own origin is unclear, and it's probably hybrid in origin. Its amino acid sequence of the nucleoprotein gene is similar to that in bird strains, so the whole thing actually may have come from a bird several years before 1918. But people seem to believe that broader surveys of bird flu strains are needed to confirm that idea. Strains descended from the last common ancestor of this clade, back here, this black-colored root on the left, have evolved at an amazingly steady rate, shown here as the number of nucleotide substitutions, or branch length on the y-axis, against year of isolation. And so both for the swine strains and the human strains, pretty much all of the points lie on something very close to a straight line, showing that flu, at least these strains of flu, do obey the molecular clock remarkably well, and therefore that relationship can be used to potentially date other sequences that may be found in the future. A very important and interesting pattern is that most flu lineages end up going extinct. What is different about the survivors? And again, we looked briefly at this story a number of weeks ago. We've seen this, a version of the same tree previously. Here again, neuraminidase and that surface protein and these colored amino acid residues that interact with the immune system and hence are antigenic, as they say in the trade. So this little table shows in the rows the substitutions in this protein in surviving lineages, that it is at the tip of the tree, still in the game, versus those that happened in lineages branched off of the stem of the tree that ended up going extinct. And it's a striking pattern. 77% of the amino acid changes in surviving lineages were in these antigenic sites, that is, the sites that interact with, that is, are seen by our immune system. Whereas in the lines that went extinct, only 47%, less than half of all the substitutions, which were larger in number. There were, 66 substitutions in the extinct lineages. Well, of course, there may be more branches, so that's forget that. That's not an important comparison because we don't know what to compare it to. But what clearly is meaningful is that a much smaller fraction of all the amino acid substitutions in the failed lineages, if you want to think of them that way, were not in these antigenic sites. More than half of them were not. That is, they were in the white-colored parts of the protein, whereas more than 3/4 of the substitutions in the winning or successful surviving line of descent were in the antigenic sites, certainly consistent with the view that as quote-unquote smart moves by the virus were a key to its survival. And in fact, this principle is used in practice to predict which of this year's strains are likely to be the ancestors of next year's. They are likely to be the ones that have new substitutions in the antigenic sites in the outward-facing proteins. And that's how they design the vaccine, which has to be made half a year or more in advance of the flu season that we hope it will defend us against. So here's a place where rather subtle evolutionary biology involving theories and a whole lot of data and fancy mathematical analyses and so on is being used to save lives by designing, it is believed, better vaccines than we would design without the evolutionary biology. At least one pandemic strain, the 1968 flu, is known to have arisen through recombination. And here again, the evidence for that is phylogenetic. The hemagglutinin in this set of strains came from a bird flu. Here, these little, is that right, the red branches? This is a tree of hemagglutinin. Yes, and so it shows recombination that is origin from bird flu, but the nucleoprotein and other genes did not come from bird flu. So that's again a historical story that could only be figured out by a lot of work involving sequencing the flu genomes from many collections from many different species, and then putting them on a tree and finding the story that best explains the distribution on the tree of the different genes for different proteins. Now finally, to SARS-CoV-2, the virus that we're living in terror of this very minute. Here's a tree from a still unpublished paper posted to bioRxiv a couple of weeks ago by Boni et al. Here's the URL that you can go to download the whole paper and read it if you'd like to, which I would warmly encourage you to do if you're at all interested. But this is the evidence that it is a bat virus. And so I've blown up this key part of the tree that includes the original sequence for SARS-CoV-2, which is a person from Wuhan, China, in December of last year. It's the so-called Wuhan human 1 SARS-CoV-2 sequence. And its closest relative is this one, just above it, which I've labeled in green as coming from a bat. It's called RA, which stands for the species of bat, TG13, etc. It's from Yunnan province. And it's very closely related to this one. Now, it's true that pangolins, that is, viruses that were isolated from pangolins, also in China, in different provinces, surround those two. Here's one below the human virus. And by the way, all of the human viruses, which we'll see a large number of in the next slide or two, they all are just a blow-up of this tip of this much larger scale tree. So all of the human ones have this bat most closely related. Then the two of them are related to this solitary collection from a pangolin, and then that group of the human ones plus this bat one and a pangolin one are the sister to this small group. Well, they're actually all identical. Pangolin isolates from yet another province in China. Okay, so you might think, well, they came from pangolins, maybe. And that is possible, that pangolins were the immediate donor. But this entire clade is entirely embedded in isolates from bats, many different species of bats, everything else in the tree except this one, which stands for the original SARS from the early 2000s, which terrorized the world when most of you were too young to be aware of it. So that's the most closely related human pathogen, and part of the reason why we call this one SARS-CoV-2 after SARS-CoV-1. Okay, so that's, again, of the same form as the phylogenies used to figure out origins of HIV and flu viruses that we just reviewed. Now, zooming down into that human SARS-CoV-2 tree, we get one that lets us tell when it entered the human population, which none of us were there to observe. How can we know that? This is a truly wonderful result of phylogenetic analysis, which, again, you can go and play with exactly as I did on this wonderful website hosted by GISAID. I'll show you their name again in a subsequent slide, but that URL is enough to take you to this incredible website, which is the constantly evolving central repository for everything about the genetics of SARS-CoV-2 and also of flu viruses, which is another story. Anyway, here's a tree that actually summarizes over 3,000 different SARS-CoV-2 genomes that have been sequenced and uploaded to this organization's website. It includes here, as this large purple dot, the original reference strain that we saw earlier, the one on the previous tree, Wuhan HU for human -1 2019. And there's one older one from 2 days previously. This was collected on December 26th, and this one on this lower branch was collected also from Wuhan on the 24th of December. And the branches are all color-coded by country, with purple for China and red for the United States, and these other greeny, yellowy colors for mostly European countries. And so from this tree, one can infer many things, including transfers, the pattern of movement around the world. For example, this group of United States strains came directly from China, obviously, right from a purple branch. This group, however, which is somewhat larger, came from Europe. And of course, they all ultimately came from China. So again, the elementary phylogenetic reasoning, once one has a tree this rich and well supported, says, well, obviously all of these lines of descent must have come from China because they're mostly present there, earliest in their history. And the date was basically December 10th. An amazing thing that we can tell just by knowing the dates of all these thousands of collections, which are indicated by the circular dots on them. Those are set on a time scale, which is here on the lower axis, showing when they were collected. And then sophisticated tree construction methods are used to place them into the tree. And the forks then represent mutations that have arisen among the sequences inside a given clade that show where its last common ancestor was located. So there it is, right? It definitely did come from China, and it first got into people there in early-ish December before anyone noticed. But we know that within a couple of weeks, people had noticed and started collecting it, and that's the origin of these earliest collections. Here's the same set of 3,000 odd sequences. It's 3,123, in case you care. That's the number shown here at the top. But in this rendering of the tree, the branch lengths, that is the x-axis, are not estimates of time, but numbers of mutations accumulated along each branch from the inferred ancestral genome at the root, which, notice, has never actually been collected, I think, although it may be, now that I look, that one or two of those under the pointer may be collections of the inferred ancestral sequence. I'm not certain. In any event, whether we have any or not, we know what it was because we have all these independently derived lineages that come from it. So it represents 0 mutations from the entry into humans. And then the branch lengths are drawn to correspond to the number of accumulated mutations along each line of descent. And of course, not all lines of descent have the same number, but there's a well-defined mean for relatively recent ones, which is something on the order of 8, and then some have more, and a few have mysteriously quite a few more than that. And then here, going back to the time-calibrated tree, just to show you it's easy to find ones from anywhere. Here's one collected on March 27th here in Utah, and it is one of those. That is 8 mutations, as it happens, from the root. Okay. These data and the analyses that we've just scratched the surface of, and a lot more information, are all freely available at the website that is hosted and maintained by GISAID, which is the Global Initiative to Share All Influenza Data, which was established in the early 2000s in order to cope with a sociology of science problem, which was that influenza virus data were scattered all over the place, and in many cases hard for researchers to obtain access to, and the workers realized this is really just a plague affecting, slowing down research that would help human defenses against influenza. And so a bunch of public spirited scientists decided we have to do something about this. Let's establish a completely transparent organization with rules that everyone must follow, and which everyone can trust to be followed, and everybody just agrees to deposit all their sequence and other key data there, and that will be a tremendous resource that will help everyone by being more open. And the model has worked spectacularly well for influenza. And although I have never seen reporting of this, it's obvious that what happened is because the people at GISAID knew how to do this from 2 decades of work with, well, not 2 decades, but 15 years anyway, of work with influenza viruses. As soon as the new coronavirus erupted, they knew exactly what to do and just established a little branch of their organization that would be devoted to doing the same thing for the new coronavirus, which they've done. In a matter of weeks, they had this up and running, and it's, it's changed the way we understand what it is that's now happening to us. And it's quite wonderful as an evolutionary biologist that we can peer in on this wonder world on a daily basis if we want, and see the picture of what we know from watching this thing spread and evolve under our noses is teaching us. It's quite remarkable, and I'm sure in a few months, the story will have many new layers, including information about how the new SARS-CoV-2 is adapting to us. So I won't take the time to go there now, but you can almost start to see from studying this tree patterns in how it is adapting and failing to adapt. One obvious one is here, this very prominent branch out of China that then gets into Europe. Notice it's exploded in Europe, but these lines have basically almost died away, as though there are adaptive mutations over here on these branches that have made them, initially rare, outcompete these originally more numerous branches, whereas others have done really well, and so on. So this is just a wonder world of data that's going to lead to, I'm confident, really wonderful and convincing stories like that, which will both be interesting as evolutionary biology and also invaluable in helping us design better treatments and vaccines and so on. Anyway, let's take a break there now, and when we come back, we'll talk about the other uses of evolutionary biology in health.